to today's event. My name is Arushi Vedya. I'm currently a PhD student at the University of East Anglia. I'm working on developing models of forebrain development using human iPSCs, and the event will be co-hosted by my colleague Sirjan, who is also an events officer and a PhD student at the University of Wyoming. So this is our event on psychedelics and the brain through research and therapy. So today we're really lucky to have three incredible speakers joining us. So we're going to start with Dr. Daniel Curtin, who's a research fellow at Imperial College London. We're then going to hear from Miss Susan Gunner, who's a holistic psychotherapist at Psychedelic Conversations. And then finally, we're going to hear from Professor Emma Robinson, who's a professor of psychopharmacology at the University of Bristol. So welcome to wherever you're joining us from. So we're lucky enough to not only have a national Women in Neuroscience community, but also an international community, because we have people joining us from all over the world, which is really, really incredible. So what is WiNUK? So WiNUK is a network founded by Lizzie English, who's our founder, and she's a PhD student at the University of Cambridge. So our WiNUK is currently made up of about 40 plus women and allies from a variety of locations. The majority of our volunteers are neuroscience undergraduate, master's, or PhD students, and we're all at different career stages. So I'm going to pass it on to Sirjan, who will introduce our first speaker.
Thank you so much, Arushi. So this is just a brief of the speakers we have today. We first have Dr. Danielle Curtin, who's a research fellow at Imperial College London. Her talk will be followed by Susan Gruner's talk and then Dr. Robinson. It is my absolute honor to introduce Dr. Danielle Curtin. She is a neuroscience research fellow at Imperial College London. Her research utilizes neuroimaging data to investigate the neural underpinnings of substance misuse and dependence. And she also looks at advanced therapeutic approaches such as non-invasive brain stimulation and psychedelic assisted therapy. Furthermore, she's even committed to including perspectives of people with lived experiences in her research and reducing bias in how research is conducted and communicated. Over to you, Dr. Curtin. We look forward to your talk.
Hi, thank you so much for the beautiful introduction. I feel like that was just a great summary of everything that I'll touch upon today. So I'm really excited to talk through my recent work with you. So let's see. Brilliant. Can you all see my slides?
Yeah.
Yes.
Great.
Thank you.
All right. Well, yeah, I guess I don't really need to give a long introduction because that was the best one I've ever had. But I will just highlight how currently as a fellow, I work through three different research groups to answer different questions about how substance misuse and dependence manifests in differences in brain function and its relationships to behavior, and then how we can target those differences using either psychedelic assisted therapy or non-invasive brain stimulation. And today's talk, I'll focus a little bit more on the role that neuroimaging can play, how it's utilized, and then how that can inform specifically brain stimulation. And to do that, I'll first start with how we even define substance misuse or dependence. So currently we define psychiatric conditions and psychological conditions with the DSM, which after the DSM-3 defines conditions symptomatically, right? Robert Spitzer was informed by Emil Kraepelin's approach. And while this may seem like a step up from the previous, more psychoanalytic way of defining diseases, unfortunately, there's a lot of limitations. So, depending on, for example, who's conducting the assessment, there's a lot of variability in how a psychiatric, psychological, or mood based condition may be defined. There's high comorbidity because a lot of conditions share symptoms. And oftentimes, if we're defining a condition by its symptoms, treatments will then address those symptoms, but maybe not the root causes. Now, a solution to this has been approached, and this can be considered the research domain criteria, or RDoC. So RDoC was established in 2009 as a way to shift from symptom-based diagnoses to biomarker-based. And this utilizes four different aspects of a framework, particularly one that I'll focus on today, units of analysis. So units of analysis describe different scales of ways to consider how a disease or condition might affect a person and its correlates and underpinnings along several different axes of, say, cognitive systems, different positive or negative valence systems, social processing, what have you. Now, currently, you can that we define substance misuse and dependence only on the right-hand side of the units of analysis with behavior and self-reports. And often, the way that we understand substance misuse and dependence utilizes the left-hand side of the units of analysis, using animal models mostly to understand cellular and circuit levels, which then are occasionally validated using neuroimaging. So, my job is to use neuroimaging to form this bridge, to develop a better understanding of substance misuse and dependence, to unite the insights we get from the more microscopic topological scale with the kind of real world experience of having substance misuse and dependence. And there's a whole host of reasons why I like using neuroimaging. Um, Not only does it provide us a kind of a more ecologically valid way of assessing biomarkers of a disease than animal models, but it can also help us identify neural correlates at a level that neuromodulatory interventions can target them, like non-invasive brain stimulation. And so today I'm going to largely focus on one neuroimaging modality, which is functional magnetic resonance imaging, or fMRI. And I do always give the caveat that unlike EEG, which gives us the summed cortical electrical activity of the brain, fMRI, when we talk about activity, it's a metabolic proxy for brain activity, right? So if I use a brain region more, in terms of there is a population level recruitment of neural engagement. It manifests an increased rate of metabolism in those areas that changes the ratio of oxygenated to deoxygenated blood, and that's what the scanner picks up. So, in doing so, we can kind of highlight already how we can look at which brain regions are involved in processes that we engage during a task fMRI paradigm. And often I really like to use tasks. You can use them to elicit different processes affected in your condition. So for substance dependence, we tend to focus on reward processing. There are so many other processes that are impacted, but for the sake of time, I'll only focus on two. And that's because we recently completed the neural correlates of reward and emotion in opiate dependent study, where we utilized two tasks. So the first task is the monetary incentive delay task, where participants could win or lose money. So this enables us to assess participants' responses to natural rewards, things like food, money, safety, shelter, sex, and sociability. And the cue reactivity task, which allows us to look at the neural correlates in response to, for example, drug-related stimuli. So in the monetary incentive delay task, this is just a super high level summary of the results that are typical of what we'd expect in people with a substance dependence. Excuse me. So we saw hypoactivity of areas associated with reward circuitry, such as the ventral striatum, also known as the nucleus accumbens, caudate, putamen, and this is during the receipt of monetary reward or anticipation thereof. We saw greater engagement of these reward regions in healthy controls as compared to people with heroin dependence. And this isn't the first study to show this. As a sanity check, we can look at other literature, which shows very similar differences in engagement of these reward circuitry. And then conversely, during the cue reactivity task, we see the opposite. We see greater engagement of reward circuitry during the presentation of drug cues in contrast to neutral cues, and these drug cues were tailored to be heroin specific for our heroin dependent population. And this also is in accordance with the literature. And so I kind of sped through that just to provide a background for why I focus on fMRI and how, in these reward processes that are commonly disrupted in substance dependence, what that can look like and how that can look different depending on what task we use. And now I'm going to focus on something that I really care about, which is functional connectivity. So if you've heard of this, you may know it is often used to assess the relationship between two or more brain regions. And just to emphasize why this is important, when we look at those pictures of blobs that are active during one condition or another, we call it sometimes blobology, because it often only looks at brain regions in isolation, and we can infer or assume a relationship between them. But without formal statistical comparison, it will remain just that. And so functional connectivity is a way to directly assess the relationship of regions over time. And you can do this in an undirected way, often through correlations between two brain regions time series. I tend to use mutual information because it assesses nonlinear relationships as well as linear. And later on, I'll also show you directed relationships where you get even more information about whether the activity in one brain region is not just related to, but is driving the activity in another brain region. And functional connectivity is really useful for us in dividing neural networks. So, outside of the context of artificial intelligence, networks have a different meaning, which is that they are a coalition of brain regions that work together to meet the demands of a specific process, right? Like a research lab uses many different people to answer certain questions within a certain theme about the brain. So too do brain regions work together to meet specific processes. And so today, because we're talking about reward systems, I'll focus often on a reward network, also called the VMN or ventromedial network. But there are so many other networks that I wish I didn't have to neglect for the sake of time today. But if you're interested in learning more about this work, it's currently under review, but you can find the preprint here. And just to give a little schematic of what we're doing, for each task, I will assess differences in mutual information functional connectivity. So that's for every unique pair of brain regions. And I will assess whether there is a significantly stronger or lower connectivity between people who are methadone dependent, so people with heroin dependence, or healthy controls. And I will also group brain regions into the networks in which they are associated. And I could talk to you about the MID task results, Q activity results, brain state dynamics, but I made this really nice summary slide to keep things a little bit more contained and just to highlight two key results, which is that despite you in the activity analysis, we show task-dependent, almost converse activation of regions within the VMN or reward network. Here we see consistently higher integration among key nodes in reward, attention, and cognitive control networks. And while that's interesting, we also saw more global decreases in connectivity. So we're starting to see a different picture emerge when we look at connectivity versus activity. And now, if you recall that this is an undirected measure of connectivity, but my fantastic master student, Brianna Austin, replicated this analysis with large-scale nonlinear Granger causality. And so the arrows here talk about the within or between networks that showed the most differences in connectivity where one is driving the other. And we saw that there was substantially higher edges with significantly greater connectivity in healthy controls versus people who are methadone dependent. And this replicates from the MID task to the Q reactivity. And this is not just within our own little lab, these results match up against the wider literature. And this is where things start to relate to the actual important thing. How does this relate to therapeutic interventions and the clinical presentation of substance dependence? So we saw in the literature, as in reference to a systematic review we recently have, so this is also under review, that people with heroin dependence typically show decreased between network connectivity. But with abstinence or a brain stimulation, so a non-invasive brain stimulation intervention, connectivity restored in the direction of healthy controls, and that related to a decreased risk of relapse or a craving. But what does that mean in the real world? This is where I really started to struggle with connecting the real world to the research lab. And so here is where the story changes tack from becoming increasingly more technical to zooming back out. Because I've started to collaborate with my colleague Koss. So he is a person with lived experience of substance misuse, and he works as a peer support worker in West London. And so together, he helped me take this image here in the review and help translate it into something that integrates more therapeutically relevant language that then I can tie to neurobiological models of substance dependence, and then tie that to the more neuroscience and network neuroscience language here. And now, as much as I want to spend more time on that, I just want to briefly highlight how over the next few years, I'll be using these better understandings of how the brain connects and works differently in people with heroin dependence to inform two types of neuromodulatory interventions. The first is that we're going to start recruiting for the psilocybin for opioid or opioid study. And I'm also working on developing computational architecture to identify personalized measures of brain connectivity and dynamics that, when non invasive brain stimulation is delivered, may attenuate some of the differences in task performance associated with substance dependence. And so, these models, if you want to learn more about them and the personalized brain stimulation architecture, please check out this review I've put up before. And as much as this is exciting, it is not all roses. There are still so many limitations we have, whether that's in how we conduct the research and recruit and retain participants, but also in our understanding of what fMRI even is. So please feel free to ask me questions about how we're overcoming some of these limitations. And if you want to learn more about my work, broadening neuroscience outside of the ivory tower and talking to people with lived experience or highlighting underrepresented researchers or people who use substances, please check out my addiction science podcast. And I just want to thank you for your time and attention, and I'm happy to take any questions.
Ask, have you always wanted to work in this field, or do you think it's something that you didn't plan and it kind of happened? Because I think a lot of the time nobody plans out their career, it just falls into place. Thank you. It was a great talk.
Thanks, Arushi. That was such a nice question. And the answer is not, I never even planned to be in neuroscience in general. My background is actually more so in public health and activism. I got really interested in academics quite late in the game, actually towards the end of my undergraduate. I was a very poor student early on. But thanks to the role of a really important mentor in my life, I kind of gravitated towards science, started asking neuroscience questions, and as I kept asking questions about the brain, they were more and more answered in the language of math. So during my master's, I began coding. I had no intention of ever being a computational person, but I was presented with the opportunity, and I'm so glad I took it despite not expecting to really engage with it. And after being computational and developing that skill set through my PhD, as part of my first postdoc, I had the opportunity to reflect and say, where do I want to apply these skills? And it wasn't to continue developing computational approaches, but to actually apply them in populations I care about, which are underserved communities and specifically people with substance dependence. I've never met an addict who wants to be one, and I really want to help people maintain a level of sobriety that they want and destigmatize substance dependence. So it came about, and I'm grateful for it.
It sounds incredible. It sounds like you can really make a lot of impact through your work, which is
Hopefully.
it's really great that you get to see the outcome, because a lot of the time if you're lab based, it's very difficult to see what you do day to day, what outcome it actually has in the real world. So that's really, really great. We had a question from Abigail, which was, how do you recruit for studies like this?
I am so glad for that question because it really allows me to touch on something didn't get to talk about here, which was how we reduce kind of historical biases in participant recruitment for experimental neuroscience. So first thing is that I don't advertise just within universities. I love how easy it is to walk into a canteen and say to undergraduates, I'll give you 20 pounds to hop in the scanner for an hour, but you're not going to find people with substance dependence that way. So I tend to partner with recovery services but I also try to partner with different faith communities. So whether that's my local churches, mosques, or temples, I tend to try to talk with faith leaders and community leaders there who tend to have a little bit of a better idea of the local community and maybe people who might be interested in reaching out. Maybe sometimes they run support groups for recovery services through there. So I'll connect with charities, local faith centers, and the NHS in order to identify participants. And in order to just finding them is one thing, but also flyer design is important. So I make sure my flyers are representative of our diversity team and the diversity and the participants we want. And last thing, I know it's a long answer, but in neuroscience, we often have a lot of bias in how we collect data towards people with curly, textured, or coarse hair. So, especially if we're doing EEG recordings, a typical EEG cap may not fit very close to the scalp on people with coarse, textured, curly hair. So, I make sure to put in my recruitment that if this is your hair type, we offer extra financial compensation because those participants often have to have extra time and effort in order to participate in the study to account for the impact that neuroimaging often has on one's hair. So we're starting partnerships with local beauticians who specialize in African hair care, who can help us do braiding in a specific way that's compatible with EEG caps. We have extra funding for these initiatives, so we put a ton of thought in our recruitment.
Incredible. I've never even thought about that aspect of it. Thank you so much. Thank you so much, Danielle. So I'm moving on to Miss Susan Gunner. So Miss Susan Gunner is a holistic psychotherapist at Psychedelic Conversations. So she has a mindfulness-based approach grounded in transpersonal psychology, focusing on trauma-informed community-centric processes that offer a broad understanding of human potential and well-being. So I've got her slides here. If Susan, if you want to
Thank you so
unmute.
much. Yeah.
Thank you.
Can you hear me, everyone? Can you hear me, Arishi?
Yeah, I can hear you and see you. Thank you. Just let me know when you want to change this.
Okay, great. I try to make it as simple as possible and hope you guys enjoy this. Thank you so much for being here and for the opportunity. So today I'll be briefly talking about the psychedelics as non-specific amplifiers, a neuroscience standpoint. And we'll dive into the three fundamental points that I wanted to share with everyone. So before we dive in, let's have a look at what psychedelics are. So psychedelics are a class of psychoactive substances that alter perception, mood, cognitive processes. They are known for their ability to induce change in thought, visual or auditory perception, and a heightened sense of consciousness. So I think you guys are already aware that psychedelics are making a comeback, and some people may say that we are actually in current wave is the psychedelic renaissance. So the history of psychedelics, they have been used for centuries in religious and healing rituals, spiritual exploration, initiation rites, especially in indigenous cultures. But the current revival of interest is in their potentially therapeutic use in medicine and therapy, based on the long historical trajectory that acknowledges their profound ability to heal the mind and the body. So we are going to look at some of these aspects. How can we use the substances to create some healing potentials? So mechanisms of action. It is known that psychedelics work, or they are the agonist of the 5-HT2A receptors, leading to altered states of consciousness, also known as non-ordinary states. And this is coined by Dr. Stan Grove. By amplifying neural communication across various regions of the brain, psychedelics can create novel connections between areas that normally don't communicate. So the experience varies from person to person, can be influenced by the environment and the individual psychological makeup. And this is where my own personal interest comes in, which I will share a little bit of my own hypothesis at the end. So let's look at the science. So science of psychedelics is undergoing a renaissance. It's a revival globally, as you can imagine. So significant research and clinical trials are exploring their potential. In universities, medical institutions, and research organizations, one of my favorite research organization or the lab that I would recommend to check out is Dr. Gul Dolan. She's been one of the most prolific pioneers in the therapeutic aspect. So groundbreaking discoveries are emerging, but how psychedelics affect the brain in treating mental health disorders and also in psychotherapeutic use. So we're going to, again, look at that at the end a little bit. So psychedelics are at the forefront of the mental health innovation, and evidence suggests that when we use responsibly in conjunction with therapy, could revolutionize the modern psychiatry. And this is exciting because as we know now, that not a lot of innovation or development happened in the last sort of probably decades, 50 years, 40, 50 years. So this is why it's exciting. So we can change the slide. So let's look at the pathways to revolution from a neuroscience standpoint again. So one of the aspects of psychedelics and I think one of the greatest findings actually was the default mode network hypothesis. And this is first discovered in the late 1990s by Marcus A. Rakel and his colleagues. So he made a huge wave by talking about default mode network. Basically what it is that it's DMN for short. It's a discovery that shaped our understanding of the brain's baseline state, meaning that it's particularly important in understanding mental health conditions like depression, where overactivity in the DMN has been linked to excessive rumination. So DMN is helping maintain order, self-identity, habitual thought patterns. This is a structured and constrained state where the brain functions in a predictable way. So we may think that this is a good idea, and this is kind of helps us function in the ordinary, waking consciousness is kind of helps us function in the best way possible. But when it comes to therapy, it's actually not so helpful. Or when it comes to mental health, when it comes to psychopathology, it's not so helpful. So we look at the entropic brain hypothesis from Robin Carhart Harris. I think this has been the breakthrough. I think this is the breakthrough hypothesis by Robin Carhart Harris. According to his theory, psychedelic states, the brain shifts into a high entropy state. Activity of the DMN is reduced. So this is why it's important to understand the entropic brain hypothesis, because, as I said earlier, DMN is not really helpful when it comes to treating mental health disorders and maintaining health and well-being. So entropic brain hypothesis is a breakthrough and really important aspect of the psychedelic use and understanding, because when the brain is in high entropy states, it is more fluid, less constrained in thinking, which can manifest as ego dissolution. This is very popular in psychedelic use, and enhance creativity and altered perception. So the loosening of fixed thought patterns can be therapeutic for people with conditions like depression or anxiety, where the brain is stuck in a rigid, low entropy state dominated by repetitive negative thoughts, such as rumination. So the reduction of DMN with the entropic brain states results in increased high brain entropic states during psychedelic experience, is thought to be the central to the effects and the benefits. So here I am showing on my slides, non-specific amplifiers, because we talk about the psychedelic substances as non-specific amplifiers, plus disarmed DMN and plus entropy, high entropy states can result in pattern disruption and potential for therapeutic insights. And this is really exciting for us to understand it from this perspective. So the other exciting discovery that I wanted to share with you, I think it's more exciting than anything we have seen so far in psychedelic therapy. And this is the BDNF and GDNF factor. So BDNF is a brain-derived neurotropic factor, and GDNF is the glial cell line derivative neurotropic factor. So BDNF is crucial for learning, memory, and overall cognitive function. And it's also, psychedelics play a role in increasing the BDNF factor, therefore it can lead to neurogenesis. So this is forming new neural connections. And then GDNF is the, acts as the protective and also helps the survival of the new cells, new neurons. So psychedelics increase BDNF expression. Research has shown that psychedelics can enhance both of these in formation of new synapses and therefore neurogenesis. And I think you guys know what that means in terms of mental health disorders. So psychedelics' ability to enhance BDNF and GDNF supports brain health, promotes plasticity, and potentially provides neuroprotective effects. And we can move on. So, and lastly, this is my favorite from the Dolan Labs by Professor Gul Dolan. So critical periods, especially coming from a trauma-informed background, I was the most excited about discovery of this now. Critical periods are windows of time during brain development when the brain is hyperplastic. These periods are often associated with early childhood and adolescence and are crucial in early life. Once these windows close, the brain becomes more stable, less responsive to change, making learning new skills or unlearning maladaptive patterns more difficult. So by the psychedelic substances reopening the critical periods allow individuals to unlearn maladaptive behaviors or ingrained emotional responses such as those seen in trauma or addiction. This relearning process is why psychedelics can lead to lasting changes. So let's look at the limitations. Of course, it's always very helpful to be transparent and really honest with some of the studies and the research and the limitations. So some of the limitations that I've listed here are complex neurobiological effects, unpredictable individual responses. People can react and respond to the substances very differently. It's really difficult to predict. Unclear long-term consequences. We still don't know, in 10, 20, 30 years of psychedelic therapy or use, we don't know how the effects will be. Challenging or distressing experiences. People are still talking about very difficult experiences, even in the most controlled therapeutic use. Generalized effect on the brain networks, lack of specific biomarkers, setting dependence, because psychedelics truly depend on the setting. And this is one of the limitations, actually. Regulatory and legal challenges, as you know, it's still very much of a sensitive area, and the research and study approvals are very limited, and it's a very arduous process. Ethical concerns about vulnerable population, of course. At the moment, a lot of people are coming to psychedelic therapy from a place of desperation, which is not ideal. And challenges in scientific rigor and research. Of course, when some of these scientific researches are difficult, it takes time, and also sometimes some of the researches are kind of led in a very sensitized environment and processes, so it doesn't give us the full results. So now let's look at the holistic view. And this is my favorite. Holistic approach in psychedelic therapy considers the whole person and their mind, body, and emotions and spirit. This model recognizes that healing is a multidimensional process and that psychedelics can affect multiple layers of the self. A holistic approach works to balance these elements to achieve a more integrated, sustainable form of healing. Can move on to the next one. So trauma-informed, community-centric approach. What does this mean in terms of psychedelic therapy? A community-centric approach in psychedelic therapy recognizes the value of collective support, shared experiences, and the wisdom of traditional healing practices. Trauma-informed care is essential to knowing how to recognize trauma responses and ensures a healthy environment. Together, they form a robust framework that minimizes risks, provides necessary structural support, accountability, belonging, and safety for the integration process. And this is why this is a very strong framework. Lastly, the phenomenology of psychedelics. So we have the research. So I believe that the science kills stigma. So very excited about the science and what is happening right now. But I cannot not speak about the phenomenology of psychedelics. And I'm going to share a quote from Ralph Metzner, PhD. He said, "The most basic paradigm shift is the recognition of the spiritual dimensions that recognized by all spiritual traditions of the world. It is not the side effects of these drugs, it is the core dimensions. It is not an add-on that you can take or leave, and that changes everything." Which means that inevitably, when we are in psychedelic therapy, we're not only looking to heal the symptoms or manage the symptoms or recover trauma, we are also having a full reset in our perception and in our connection to self. That changes everything. And lastly, I would like to leave you with another piece of quote from David Nichols, PhD. So David Nichols was a chemist in the early research, and he produced LSD, DMT, MDMA for studies undertaken by MAPS, Dr. Strassman, and John Hopkins. And he said, this is really related to the neuroscience, and this is why, especially for the purpose of this conversation. He said, where the 2A receptor is expressed is on the cortical cells, which are the major computational units in the brain, where all the unconscious material processed and goes back to single-celled organisms. It's an ancient receptor. Nature found something that worked really well, so it's kept it all the way up through the evolutionary chain. I really love this quote from him. And lastly, my hypothesis. So it's an unconventional approach, and especially trauma-informed therapy with psychedelics. And again, I would like to tie it down with Dr. James Fadiman, one of our pioneers in the specifically microdosing realm. And he said, don't take LSD to learn rocket science. Learn rocket science and then take LSD to open up much greater possibilities for expansion. So therefore, I would say that my approach to psychedelic assisted therapy is very similar. I'm very passionate about building and developing preparation, maybe models as a foundation, and then bringing in the psychedelic therapy, not the other way around, because sometimes out of desperation, people are indulging in these substances prematurely, and that's why we are also equally experiencing re-traumatization, difficulties, and instead of benefits, we are currently at the moment seeing a lot more negative effects as well. So let's be responsible, conscious about how these substances work. They are non-specific amplifiers, meaning whatever it is contained and suppressed and carried in the individual as a container. You are the container. And it's not that the psychedelics are taken and the substance is working with us. It's actually the other way around. We are working with the substance. So we are responding to the substance based on where we are in our consciousness, where we are in our progress. And that is my hypothesis. Thank you so much for your attention. Please check out the Psychedelic Conversations podcast for more interviews with these amazing teachers, elders, facilitators of our time.
Hi, I just wanted to kind of have a comment to say that I was really glad at the end you mentioned about making sure that we're not using psychedelics in the first instance, making sure people have the coping skills and the grounding strategies. I was say that was my question of kind of how much of the pre-work you do, but I'm glad you brought it up. As I say, as a neuroscientist and a future psychotherapist, yeah.
That's a great question. I think we're very, very new. Everything we're developing, we're still in the process of developing different tools, modalities, maybe foundational structures that we can help people to create that fundamental, baseline first. I think that's going to be depending on the assessment of individual, depending on individual where they are and how sensitive what they are experiencing, depending on the struggle, the challenge, mental health challenge. I think it's going to have to case by case, individual by individual. But I wouldn't say a time frame, but I think, like I said, it's going to have to be coming from the professionals who can discern and assess properly, that can provide that kind of, initial entry, then the psychedelic therapy incorporated. Hope that helps.
Yeah, no, thank you. As I say, it's really reassuring that you're working like that.
Thank you.
There's a question from somebody called Nada Salah in the chat box. So some experience deterioration in their cognitive abilities and memory. Is it a direct effect of the psychedelics or the illness? How do you assess that, e.g. Bipolar or PTSD?
Can you read that again, Arishu? Sorry, I missed the middle bit.
Sorry. So some people experience deterioration in their cognitive abilities and memory. So is this a direct effect of the psychedelics or is it the illness? And how do you assess that? So basically, how do you differentiate between the effects of the mental health issue or the drugs?
Yeah, this is great. So I think one of the biggest challenges is going to be that we're never going to know until we develop, really robust, structures and understanding the medicines. I think we still, again, I don't want to repeat it, but we are at the early stages. But one thing that we know, and which is my passion, I'm really passionate, is I have this understanding that maybe microdosing could be far more beneficial initially for those that are suffering from those kind of mental health disorders. Microdosing initially would be so much more helpful. And we have seen, we have a lot of research studies out there now that, MindBio has done, I think, 3 phases of microdosing LSD. And it's very promising for any cognitive decline or those kind of disorders. Thank you for the question.
Thank you. I just had a quick question. Basically, because obviously psychedelics are so controversial, there's also this thing about like pharma companies having a financial incentive in investing in them. So do you think that if pharma companies made equivalent synthetic compounds instead of natural psychedelics, do you think that would spread more awareness about psychedelics? It's a bit of a confusing question, but
I know it's a really good question. Yeah, it's a really good question, Arushi. I mean, my colleagues and my friends were always having these discussions, right? What are the biotech companies are doing actually? Is it for the money? Is it really they're trying to help people? I mean, I was at the Psychedelic Conference, Colorado, and I met some of the most amazing biotech people from biotech companies. One of them was Daniel from Germany, and he said they were creating a novel pills from ketamine that people can take it at home. and it proves to be working. And especially, I think, again, it comes to the region and the, what part of the world other people are. Some people are really open to the full-blown psychedelic, full spectrum experiences where it induces, mystical experiences, and they're happy to have that experience. And some people are very careful, very logical, and they really want to be safe. And they may choose the biotech route. So we don't know. There's so many different interests, but one thing is for sure. I think with the level of technology and understanding, I think some of the novel ones are going to be really helpful. Especially, I think they did one with research with a novel drug without the psychedelic effect, and it was helpful with dementia. So we're going to see some of that coming up as well.
Perfect. Thank you so much. Just for the interest of time, I think we have to move on to the next speaker. But if anybody has any questions, we can answer them at the end. Thank you.
Our next speaker is Professor Emma Robinson. She completed her bachelor's and PhD at the University of Bristol and worked as a research fellow at the University of Cambridge. Her research investigates the mechanisms which regulate normal and pathological emotional behavior and their interactions with antidepressants. She also runs a research program in animal welfare and is the academic lead for the animal facility. We're very happy to have you. Thank you.
Thank you for that. Hopefully you can see my slides.
Yeah, it's perfect.
Yes.
Brilliant. Okay, so I've sort of split this into two bits really, because I have a quite unusual career path into working with psychedelics. I certainly didn't set out in my career thinking I was going to be working with psychedelics. And I also have an interest in animal welfare. So I'm going to talk a little bit about my career and then finish off by just a snapshot really of some of the work that we're doing in this field. So this is just an overview of what I think is a slightly unconventional career path. And I think, well, hopefully it's an example that often you don't necessarily start out thinking you're going to be doing this and then end up down a different path. So let me just put my laser pointer on. So I actually started out with an ambition to be a vet, and you might see a bit later that an interest in animals and animal behaviour has been at kind of the heart of my passion, really. And I didn't get in. I didn't get good enough A-level results, and I didn't really quite know what to do. But Bristol University kindly offered me the opportunity to come and study a degree in this subject called pharmacology, and it was a route I saw to getting into veterinary medicine. So I thought I'd do that. So I came to Bristol, had a great time studying pharmacology, learning all about drugs and how they act within the body. And I started to identify this area of neuropharmacology where we were studying and learning about how drugs affect the brain. And I was just intrigued, and I really was taken by how little we didn't know. And then I had this very interesting period in 1995, end of my degree. I got to do a research project, and it was great to hear about the addiction work earlier. I started my research career looking at alcohol withdrawal syndrome in mice. And kind of two things really struck me. Firstly, that you could use an animal model to study what I had thought of was such a human condition. And we got some really interesting data, but also just how much I really enjoyed it and how much I found that kind of the unknowns of neuroscience drawing my interest. And at the same time, I worked at a local vet's where I was doing some work experience, and I really didn't enjoy that. I was surprised by my passion for the kind of veterinary side had really changed. I decided that actually I really wanted to stay in science. So I took the opportunity of a PhD, and the PhD that was available that really took my interest was to study the mechanisms underlying antidepressants. And particularly I was interested in the delayed onset of action. And you've heard a little bit about that already, but there's many challenges in how we treat mental health, but one of them is that the drugs that we currently use, they don't work for everybody, but also they can take many weeks before they have an effect, and that comes in a bit later with the psychedelic therapy. However, there was a real irritation for me at this stage, and that was that the animal models that were being used to study antidepressant effects just didn't feel to me like they were aligned with what was going on in the human condition. And I couldn't find a postdoc that I really liked the sound of, but I was really enjoying teaching. I'd been doing some temporary roles teaching. So I took a temporary lecturer and off I went and thought I was going to do teaching and admin. And that's pretty unusual in my field to go off out of research for as long as I did. So I was out of research for 6 years. But then I kind of had a bit of a change of heart and realized that there was a route to which I might be able to come back and answer this question. And it was that I could use my interest and expertise in animal behavior to perhaps make better animal models to study the antidepressant effects. And I was very lucky that in 2006, I was able to convince some funders and the University of Bristol to support me in an academic fellowship in this sort of subject of integrative pharmacology, to try to achieve that, to try to come back and revisit the field of depression and look to develop new animal models. And at the same time, I also managed to get a place to take my visiting fellowship to Cambridge, which was absolutely fantastic. So I learned psychology, which I'd never done, spent a year learning psychology. And then I came back to Bristol and I set up my independent research lab with my fellowship funding. And it's kind of led to, this is my kind of core area, really. So I have a primary research ambition, and that is to establish novel tasks that allow us to understand the mechanisms that underlie depression, and also developing better antidepressants. And that really led to one real core question: how do you quantify emotions in animals objectively and with clinical relevance? And that's not really a very easy question. Lots of things come up. Do animals have emotions? As we've already been hearing, lots of the stuff that we've heard is all about the kind of the very human characteristics of how psychedelics work. Rumination, how they change our perceptions, default mode networks. These are all things that you might not actually think you could even model in animals. And I hope I'll give you a bit of an insight into how we might be able to at least look at some aspects of these symptoms in humans. And then the other thing I also started to question was how normal laboratory rats and mice really were. And that led to an interest in psychiatry, animal behavior, ethics, and laboratory animal welfare. And that's now the core of my research. So I thought I'd also, just because lots of people talk about their research careers, well, I'd just tell you a bit about my work-life balance, because it's a mixture of everything, and everything informs everything, really. So I run research programs in psychiatry and animal welfare. We were really lucky. We were a well-funded group. We've got lots of really great people working in the group. I'm in the academic lead for the animal facility, so that's my kind of main leadership role within the university. I've launched a welfare initiative, which is all, we call it the 3 H's: housing, handling, habituation. It's all about improving the welfare of laboratory animals to make better science, and also to benefit their welfare. I love doing public engagement in science, and this is kind of, we heard a little bit about people with lived experience. I found it one of the most fantastic ways to learn about mental health was to go out into the public, talking about our science and hearing people's stories as they came up to talk to us. And then for that kind of work-life balance, because I really do believe that kind of outside world, that ability to get some space, is really, really good for us. And for me, being an academic, if you're not careful, you can get very buried into your science and your lab. So I bought a small farm about 10 years ago in Devon. It's a very derelict farm, and I built a log cabin. It's my log cabin that I live in at weekends. This is a view of the farm. It's really beautiful. It's got some old buildings, and I spend my time with a couple of lurchers. My niece lives with me there, and we've got horses. And I also, in Bristol, run a not-for-profit dog training club, which is, again, my animal behavior. So I do lots and stuff, and I reckon a good work-life balance is to be really busy in absolutely everything that you're doing. Okay, so let's talk about psychedelics. So I'm really interested, and this is a very specific question I have: how do these psychedelics induce such a rapid and sustained antidepressant effect? And we're talking about a whole range of really interesting compounds. So we've heard about these as being serotonergic 2A agonists. They actually act on quite a number of different receptors in the brain. We've got substances like DMT. Sorry, these are the wrong way around. That should be psilocybin. That's DMT. So this is the psilocybin mushroom. This is ayahuasca, LSD, synthetic, and then mescaline. And these all induce these very profound effects on the brain. And we know from all the clinical data and the work to date, and this is the results from a recent clinical trial. So this is the phase 2 clinical trial from one of the companies working in this field. This is a synthetic version of psilocybin called COMP360. It's now in phase 3 clinical trial. And what makes psychedelics so intriguing to psychopharmacologists like me, as well as all the other people who are interested, but my real interest is, how is it that you can give this drug where its effects only last for a few hours? So when people are given psilocybin, the effects last for a few hours, and then there is this sustained improvement in mood, and it lasts for months. So this clinical trial went out to 3 months, and you can see that these patients show a rapid improvement, and then it slowly starts to come back. So it's not forever, but it seems to make people well for a really long period of time, and way after the drug has been metabolized and excreted from the body. So how is it that this could be happening? And we've heard lots of really interesting ideas about the different hypotheses. I think at the moment, we really don't know how psychedelics work. We have lots of studies going on at the molecular cellular level that I'm interested in, but there are challenges to that, the models that we use, but also the doses we use. So a lot of the effects that are reported in animals use very high doses of these drugs, and probably generating effects that may not be happening in people. So my interest really lies in this, and I have a kind of big picture question, which is what is mood? And then my specific questions is what are the mechanisms that underlie mood? And I want to address that by using animal models, because I think that allows us to simplify our questions and also to integrate the cellular molecular processes right through to behavior. But actually, I don't know what mood really is. So I asked Chatbot, because these days everybody should ask Chatbot to see what Chatbot said. And Chatbot told me that mood is a complex interplay of biological, psychological, environmental, and social factors. And that really fits very nicely with an area that we've been interested in for the last sort of 20 years. And that is these neuropsychological mechanisms and mood. And what that relates to is this idea that core to mood is past experience and future expectations. And these are all generated from memories. And so a lot of what patients with depression are experiencing is that these past memories have very strong and very negative influences on their current and future expectations. And that can also underpin some of the symptoms we think of, like loss of interest in rewarding activities, rumination of these negative memories. And when we look at this, it's very hard to identify a biomarker of depression. But one of the things we have been able to identify is that people with depression seem to have a fairly robust and consistent negative affective bias. So this is where their mood is influencing their underlying cognitive processes. And in particular, what has been noted is that this negative bias is influencing learning and memory. And we think this could be contributing to their low mood. And so what we've been interested in is, could we be looking at, and could these rapid acting antidepressants, the psychedelics, could they be acting by an acute modification of affective biases? And could it be this acute modification that then enables this sustained improvement of mood, i.e. it modifies these affective biases, learning and memory processes, so some sort of very specific neuroplasticity change? And this is how we've gone about trying to answer that question. So again, this is a much more simplified approach. It's very different to what we've been hearing about in terms of the clinical studies. But what we've generated is an animal model of a negative affective bias in depression. So it's only taking one aspect of depression. It's only looking at one component, but it allows us to take what is a translational behavior, so something that we know happens in people, and take that back to a rodent model, and then ask that model to tell us more about the underlying biology, and particularly how does that model interact, how does psychedelics interact with that model? So to give you an idea of how we actually do that, if I just play my video, hopefully it'll play. So the video, this just shows you a simple video of how the task works. So this rat has been given 2 bowls to choose from. I'll play it again. He's been given 2 bowls. They've got different substrates in. One of the bowls has food in it and the other one doesn't. And he's learned that the substrate predicts where to find the food. And that essentially forms a memory. And so what we do is we train the animals to learn about, we teach them 2 specific memories. One is formed in a negative emotional state, the other one in a neutral state. And together, this then generates a negative affective bias, which we can quantify by asking the rat, which did you prefer? So the animal on this day has a negative affective state and learns that this has the food in it. The next day, neutral state, this has the food in it. Now, these are equal values, so they should come out 50-50. There is no reason why the rat should prefer one or the other than due to this affective state manipulation. But when we do the choice test, that's exactly what we find. The rat shows a preference for this and a bias, a negative bias for this, and that's quantify. So the great thing with these kind of studies is we can put a number on it. We can quantify And so what happens is we can quantify a negative affective bias, and then we can look at what happens if we administer a psychedelic before the choice test. And we can see how does that work both in the short term and at 24 hours. And we also have another version, which I'll show you some data from, which is where we actually gave the psychedelic here. So we actually said, how does the psychedelic change your affective state? And these are the data. And I put in here also ketamine, because actually what's really interesting is that although we've talked mostly about the psychedelics, so these are serotonin psychedelics, these are drugs which induce their hallucinations and their altered perceptions through affecting the serotonin system. There's also a drug, ketamine, which is a dissociative anesthetic, which at low doses has been shown to be a rapid-acting antidepressant. It's used clinically. It's licensed in the US. It's used in the UK in clinics. And we've used both of these drugs in our studies because we've been able to sort of triangulate to see if similar effects are seen with both of these. Maybe this is like a common underlying mechanism. So these data relate very much to what I've just shown you. This would be the negative bias that the animals have experienced. So this is a negative affective bias generated by a drug that causes the animal to feel a bit anxious when they're experiencing it. And then when we give psychedelic treatment, so this is psilocybin, quite low doses, so these are clinical doses, you can see that the animals now don't retrieve that memory with the same negative bias. In fact, it's neutral. And the same with the slightly higher dose of psilocybin and also with this dose of ketamine. So these are all clinically relevant doses. Now, because we've given this acutely, and of course, as you know, the psychedelics could be inducing this effect where the animals maybe just don't know who they are or what they are, and therefore we might be seeing this just because they can't perform the task. So what we also do is a control test. So this is where we haven't generated a bias with an affective state. We've generated a bias using a change in reward value. And so it's a slightly different, but it's a very good measure of their memory. And they're absolutely brilliant at it. They can still all do the task, no matter what treatment we've given them. So this tells us that acutely, these drugs are able to induce this very selective attenuation of negative affective bias, so quite a specific effect. What we then did is tested the animals 24 hours later, and we were hoping to see something like this. We were not expecting to see what we saw. And that is that something quite strange happened. So here is the same negative bias 24 hours later. But when we've given the psilocybin treatment or the ketamine treatment, these animals are now retrieving this memory rather than the negative bias with a positive bias. So quite different, a quite different outcome to what we predicted. And really, the only way that this could have happened would be if there'd been some sort of relearning of that negatively biased memory. So these are reward memories, so it's not like we've changed the bad memory to a good memory, but what we've done is we've taken a negatively biased rewarding memory, and where the animals would normally have this negative bias after the psychedelic treatment, they seem to have been able to relearn it with a more positive affective valence. And that ties in really interesting. It's quite interesting to talk to people who've experienced this treatment with their depression, because they talk about the fact they can still remember traumatic experiences, but the emotional component just doesn't feel quite as strong, and they feel like they can cope with thinking about them more easily, which is, I think, really interesting. And again, it's similar between psilocybin and ketamine. And then we also looked at what happens. What does psilocybin actually do itself when we ask them to form new experiences? And what's really interesting is this is where they're different. So psilocybin is different from ketamine. Psilocybin can induce this positive bias in new experiences, not at the high doses, but at these lower doses. And that actually looks surprisingly like this drug. And this drug is a conventional antidepressant. So this is venlafaxine, which is prescribed for depression at the moment. But interesting, not ketamine. So there are some differences, but there seems to be this common mechanism. And so what we've done is we've proposed this hypothesis, and we published this quite recently. So we've talked about this as turning the glass from half empty to half full. And we suggest that there may be an interaction between the biological effects of the drug and then experience-dependent mechanisms, so this sort of selective plasticity, which could explain their rapid and sustained effects. So here's some bit of pharmacology. So ketamine is here, and this is psilocybin. And what we know by zooming in on the neurons in a particular part of the brain, this is the cortex, where we know there's lots of these 2A receptors, but also there's other, there's parts of the brain really important in emotional regulation. And this is another drug that has antidepressant effects, scopolamine, I haven't talked much about, but it also works here. And what we think happens is that these drugs are all able to what appears to be at these low doses, we get this rather selective modulation of this circuitry. We get a shift in what we call the excitatory inhibitory balance. So it changes the balance of the neurotransmission, a bit like what we were hearing earlier about this changes in cortical activity, the disintegration. We think that this could be underlie these effects could underlie that exact thing that's observed in imaging studies in patients. But what we've suggested that is perhaps slightly different is, rather than this all being about driving plasticity in a kind of non-specific way, we think actually the plasticity part of it comes in this sort of phase 2. So phase 1 is the sort of selective modulation of the affective bias circuit, modulating these mechanisms of emotional regulation. And then in phase 2, we've got this experience-dependent learning and memory. And we've actually done a few studies, I haven't shown them here, but we've done some studies to suggest that there is this direct interaction with learning and memory. It's dependent on protein synthesis, so that sort of fits with plasticity and new synapses forming. And it's also very drug and dose dependent, which again fits with our idea about selective toxicity. And then what we're suggesting, the glass half empty to glass half full, is that negatively biased memory is then enabling that to be reactivated during this therapeutic window, which can then be effectively relearned as a positively biased memory. And if you can shift that balance in the brain from lots of negatively biased memories to more positively biased memories, then that should, coming back to what I was saying at the beginning, underlie a sustained improvement in mood. So that's the kind of ideas that we've been working on. Okay, I've only given a really brief indication of some of the data, but we've got lots of other sort of molecular work and more behavioral studies where we've been able to bring that sort of evidence base together. And I think the other thing that's been really interesting with this is working with an animal model. We are not really able to sort of align that to some of the more human ideas around sort of the religious experience, mystical experience. We think this is really functioning where we are actually able to directly show this modulation of these circuits of the brain that modulate our emotions. And I'll just finish off by thanking all the people that have been involved. So lots of really great people who work in my lab on the projects, our funders, and particularly Compass, who we work with for the studies on COMP360, and Boehringer Ingelheim, who we work with the ketamine study, and our industrial collaborators. And I will end there.
Yeah, thank you so much. That was really, really enjoyable. I really liked your career journey, and especially how you showed things that you do outside of the lab also, which is really, really great. Hand it over to Sirjan, who'll discuss the blog.
Yes, so we also have a WiNUK blog where we have a recent series that includes preparing from your undergrad to your postgrad pathway. We talk about research, we talk about review articles. The most recent one is on psychedelics, which I authored, where I walk you through the ancient rituals to modern day applications of psychedelics. So feel free to check that out. The link is also in the slide. Okay, Arushi, you can change the slide. Furthermore, we'd also be very, very grateful if you guys could stay in touch. So sign up as a member on our website. The socials are also linked. We have an Instagram, a Twitter account, an Instagram, and all the socials are linked. Please do not forget to fill the feedback form. We will put it in the chat once again. And thank you so much for joining. Have a lovely evening.